ఓకే. రజనీ మేడం గారు
సార్
మీరే
టెల్
స్టార్ట్
మీ సార్.
చేయాలి. మీరే స్టార్ట్ చేయాలి.
వెయిట్ సార్, స్టూడెంట్స్ జాయిన్ అవుతున్నారు. షెల్ వి స్టార్ట్ సార్?
ఎస్ యూ మే ప్రొసీడ్.
Good evening, everyone. Good evening, sir. This is Dr. A. Rajni, Head Department of Botany and Food and Nutrition. Today we will start to the faculty exchange program for the College of SRR Government Degree College, Karimnagar. Students to listen to the class today. Sir name is Dr. Padala Tirupati sir. Assistant Professor in Botany in SRR Degree College, Karimnagar. Today onwards, 1 week, this program will start today. This program will start today, and 1 week program is there. Your totally 25% syllabus to complete the sir. Okay, students, all the best. Thank you, sir. Welcome to the session.
Thank you very much, madam. It is my immense pleasure to interact with our beloved friends of prestigious institution of our RBVRR Women's College Autonomous, Hyderabad Indian City. So this is Dr. Padala Tirupati, Assistant Professor, Department of Botany, Food Science, Crop Production. Today onwards, I will going to interact with you in plant physiology, one of the most important area, that is nitrogen metabolism, nitrogen fixation, and the plant growth regulators, that is phytohormones, growth promoters, and growth inhibitors. We'll going to discuss today onwards. So, my dear friends, with your permission, madam, I will start today's class.
Okay, sir. Continue,
Okay.
sir.
I hope my PPT is visible.
Yes, sir.
Okay. So, in this today, my dear friends, I am going to interact with your wonderful day. That is an overview of nitrogen metabolism. We know that nitrogen is one of the important macro elements or macronutrients for our growth and development, and also so many physiological activities in our day-to-day life activities, including animals, plants, and human beings. So nitrogen metabolism, nitrogen is also one of the most important for all around development of the plant growth development, at the same time even the seed germination. సో ఇది ఎన్లార్జ్గా కనబడుతుందండి పెద్దగా స్లైడ్ పెద్దగా కనబడుతుందా చిన్నగా కనబడుతుందా?
Sir, make it slide share.
స్లైడ్ షేర్ లో పెద్దగా కనబడుతుంది ఇప్పుడు?
No, sir. It's not slide share. చిన్నగానే ఉంది.
చిన్నగానే ఉందా?
Sir, my colleague to join Alekia, I will be come to 10 minutes with him.
ఓకే
Come back 10 minutes.
ఓకే. ఇప్పుడు కనబడుతుందండి పెద్దగా?
లేదు సార్.
పీపీటీ ఎందుకు పెద్దగా కనబడట్లేదు ఇప్పుడు?
స్టూడెంట్స్ మీకు కనిపిస్తుందమ్మా?
చిన్నగానే ఉందా పెద్దగా? చిన్న మిగతా స్లైడ్స్ కనబడుతున్నాయా?
సైడ్ కి స్లైడ్స్ కనిపిస్తున్నాయి కదా మనీషా?
అట్లా కనబడకుండా పెద్దగా వస్తలేదండి?
రావట్లే కదా అమ్మా?
1 సెకండ్ మళ్ళీ.
నో సార్, ఇట్స్ నాట్ కమింగ్.
ఓకే ఓకే, మీరు అలాగే ఉండండి, నేను మళ్ళీ అసలు బిగినింగ్ నుంచి వస్తాను.
సార్ పర్లేదులేండి సార్, స్టార్ట్ చేయండి. ఇది 40 minutes కదా మళ్ళీ.
గురించే రెడీగా ఉండి. ఇప్పుడు రాలేదు కదా?
నో సార్.
ఇక ఇలాగే కొనసాగిద్దాం.
ఓకే ఓకే.
సో వి నో దట్ nitrogen metabolism is one of the
The
most.
screen is not visible. Your desktop is visible, sir. PPT is not visible.
Visible అయితలేదా? ఇప్పుడు
No, sir.
ఇప్పుడు
No, sir. Now.
వచ్చిందా మేడం?
No, sir. Now okay, sir.
ఓకే. ఎన్లార్జ్ అయిందా? అలాగే చిన్నగా ఉందా?
అలానే ఉంది, బట్ కంటిన్యూ చేయండి సార్.
ఇట్లాగే కంటిన్యూ చేద్దాం. ఓకే.
చేయండి.
Okay. So in our day-to-day life activities, we observed so many growth regulators. At the same time, the micronutrients for the development of the plants and also the all physiological metabolic activities. The plants, they are depending upon the different nutrients. We know that nitrogen is one of the most abundant element in the entire atmosphere, which is available more than 78% of the 78% of the atmosphere gas, that is molecular nitrogen. But even though the molecular nitrogen is not utilized by our plants. ఇది నేను రాసేది కనబడుతుందండి?
ఆన్లైన్ లో మాత్రమే చెప్తాడా? ఏం చెప్తాను?
స్నేహలత,
ఎవరో తెలియదు. మేడం చెప్పిస్తారు.
స్నేహలత, నేను రాసేది కనబడుతుందా? స్నేహలత. Okay. So the entire nitrogen metabolism broadly classified into two types. The metabolism of the nitrogen, that is one of the catabolism. So catabolism related to the degradative process, the concern with the breakdown of the complex molecules into the simpler manner with the release of the energy. The second type of the metabolism, that is anabolism. Anabolism related to the biosynthetic reactions which is involved into the formation of the complex molecules from simpler precursors. So these two types of metabolic activities or metabolism which we are observed in the all living systems including plants. Next slide కనబడుతుందండి? Next slide వచ్చిందా? Anybody can respond, my dear friends.
Yes.
Okay. So we know that nitrogen metabolism, which is available in the atmosphere, it is a major attributes of organisms such as functions and structures. Major operations and mechanism of metabolic pathways is provided by the proteins. The nitrogen is one of the major component in the amino acids. These amino acids leads to the formation of the proteins. So even though the genetic information is stored in nucleic acid polymers, which is depends upon the nitrogen, and each of the monomer of these macromolecules has an individual metabolic pathways. So such as metabolic pathways are, we know that already two types, that is anabolic and metabolic pathways. So then what is the process, which type of processes involved in nitrogen metabolism? The most of the prevalent essential macro elements which regulates plant growth, especially in agricultural systems. Have we ever observed so many plants related to leguminaceae plants? ఎవరైనా లెగ్యుమినేసి కుటుంబానికి సంబంధించిన మొక్కలను చూసారండి మీరు ఎవరైనా? ఎనీబడి ఎల్స్, మై డియర్ ఫ్రెండ్స్? Okay. So even though the non-leguminous plants also we are observed so many different plant or root nodules we are observed in the process of the fixation of the nitrogen from atmosphere. So the major source of the nitrogen which is available in the atmosphere gas atmospheric gas is not unable to utilize these plants because of that is a complex, that is a molecular state of the nitrogen. The higher plants unable to utilize due to the mechanism, nitrogenase enzyme and NIP genes, which are not available or not present in the higher plants. So these plants are require higher amount of the nitrogen as it is important their structure and metabolism. Why? Because the structure is also depending upon the nitrogen and also the functions, majorly the osmoregulatory process and metabolic activities depends upon the nitrogen molecule. So that's why the molecular nitrogen is unable to utilize these plants. That's why the plants are major source or depends upon the simpler form of the nitrogen or nitrogen oxides or nitrogen usable nitrogen forms. But unfortunately, most of the plants cannot utilize in this elementary form. So they have to depend on the soil and they acquire nitrogen inorganic form either as ammonium compounds or nitrates. So these are the different forms of the nitrogen: nitrates, nitrites, and ammonia and ammonium. These are the different forms of the nitrogen molecule. Am I audible, my dear friends? Am I audible? Slide is moving. ఈ స్లైడ్ కదులుతుందా? మీకు స్లైడ్ వచ్చిందా? Okay.
Yes, sir.
Then nitrogen metabolism including both anabolic and catabolic process. Then anabolic processes are, what are anabolic processes? We know that the first anabolic process of the metabolism in nitrogen metabolism, that is nitrogen fixation. Then the second anabolic process, that is amino acid synthesis. Then third anabolic process of the nitrogen metabolism, that is the protein synthesis. We know that protein synthesis contains two different steps, that is one is transcription and second one is the translation process, which are regulated by the involvement of the nucleic acids, that is the DNA and RNA. In nucleus part, the transcription process we are observed in the cytoplasm, the translation process we are observed. So these three includes anabolic processes, and catabolic processes also contains three different processes. Number one, the proteolysis and amino acid destruction. Then second one, denitrification, and third one, nitrification. These three processes are the catabolic processes. I'm sure the in nitrogen metabolism includes how many processes? Two processes: anabolic processes and catabolic processes. Anabolic processes are nitrogen fixation, amino acid synthesis, and protein synthesis. Then catabolic processes are the proteolysis and amino acid destruction, denitrification and nitrification. Am I clear, my dear friends?
Yes, sir.
Good, very good, Banu, very good. Then the nitrogen fixation. What is what we are observed about the nitrogen fixation? What do you understand about the fixation? Fixation means the molecule or nutrient or the molecular state of nitrogen which is available in the gaseous form in the atmosphere, which is from molecular state into the usable form of the different nitrogen forms into the soil by the physical process, by the chemical process, or the involvement of the other microorganisms. That is nothing but we can call as biological nitrogen fixation. So here in this slide, we are observed the process of the nitrogen fixation. The molecular nitrogen, which is convert into ammonia. Very, very important, my dear friend. The principle, I hope the red color principle you are visible. Am I audible? The molecular nitrogen is converted into 2 moles of the ammonia, which is utilized 8 protons, 8 electrons, and 16 ATPs. Come on, anybody tell, my dear friends, how many protons are needed for conversion of the atmospheric nitrogen into 2 moles of ammonia? How many protons? Come on.
8.
8 protons. How many electrons?
8.
8 electrons. How many ATPs are needed?
16 ATP.
So 16 ATPs are utilized in the process of nitrogen fixation from the atmosphere. During the fixation process, it is converted into 2 moles of ammonia from molecular nitrogen, and 16 ADPs are produced and 16 inorganic phosphates are produced. So in this process, the most important enzyme is involved. That is nothing but nitrogenase enzyme. Which enzyme is needed for conversion of the nitrogen, molecular nitrogen into 2 moles of the ammonia, my dear friends? Nitrogenase enzyme. So nitrogenase enzyme, which is involved for conversion of the molecular state nitrogen from the atmosphere into soil nitrogen or the usable forms of the nitrogen, or that is nothing but the ammonia, nitrites and nitrates. So 16 ATPs are needed, 18 protons are needed, and 18 electrons are needed. So the utilization of the 8 protons and utilization of the 8 electrons and utilization of the 16 ATPs. So that means it is what we can say that the energy, ఇది శక్తిని వినియోగించుకొని ఇది మనకు 2 moles of the ammonia గా కన్వర్ట్ చేయబడుతుంది. The conversion of 2 moles of the ammonia, the utilization of the energy. How much of ATPs energy needed? 16 ATPs. So this result into the 2 nitrogen atoms are converted into the 2 moles of ammonia. Okay. Then this ammonia further formed is used for the synthesis of amino acids. We know that 21 essential amino acids are there now. 21 amino acids. So amino acids biosynthesis, which are from 2 moles of ammonia, which are converted by nitrogenase enzyme from atmosphere. Am I clear? Am I clear? And my dear friends, remember the nitrogenase enzyme, which is useful for the destruction of the catalyzed into the broken down of the 3 hydrogen bonds between 2 nitrogen atoms. 2 నైట్రోజన్ పరమాణువుల మధ్యలో ఉండే 3 హైడ్రోజన్ బంధాలను బ్రేక్ డౌన్ చేస్తుంది, విచ్ఛిన్నం చేస్తుంది. ఏది? నైట్రోజనేజ్ అనే ఎంజైమ్. సో నైట్రోజనేజ్ ఎంజైమ్ which is useful for breakdown of the hydrogen bonds in between 2 nitrogen atoms. So this is the mechanism of the nitrogenase enzyme. So by this process, the nitrogenase enzyme utilize 16 ATPs, 8 protons, 8 electrons. Am I clear?
To respond, ma.
Yes, sir.
Okay, very good. Snehalata. Then the biological nitrogen fixation. Actually, biological nitrogen fixation is carried out by highly conserved complex of proteins, nitrogenase complexes by the prokaryotes, especially bacteria and cyanobacteria. These are the prokaryotes and microorganisms are converted of the atmospheric nitrogen into the soil nitrogen. Why? Because the special type of mechanism, the nitrogenase enzyme and NIP genes are present only the prokaryotic organisms. So that's why only the biological nitrogen fixation involvement of the organism only prokaryotes. Cyanobacteria, that is nothing but blue-green algae, or cyanophyceae, algal members only responsible for the fixation of the nitrogen from the atmosphere, and also some of the bacteria, nitrogen fixing bacteria, that is what we can say that the bacillus species and some of the one of the most important bacteria, that is
Nitrobacter.
that is responsible for the root nodules. So rhizobium leguminosaurum bacteria and some of the symbiotic bacteria, some of the asymbiotic bacteria also responsible for the fixation of the nitrogen from the atmosphere. So nitrobacter and thiobacillus denitrificans. These are the bacteria are involved from the involved in the nitrogen cycle. But thiobacillus denitrificans bacteria, which is destruction process of the nitrogen, which is harmful to the plants. Why? Because the usable form of the nitrates into converted into molecular nitrogen. So different bacteria are involved in the process of biological nitrogen fixation. And my dear friends, here the electrons are transferred from pyruvate to denitrogenase via pyridoxine or flavodiazine, and the denitrogenase reductase enzymes also involved. And the denitrogenase denitrogenase one electron at a time with at least 6 electrons required to fix one molecule of nitrogen. So one molecule of nitrogen is used the 6 electrons, and in additional 12 electrons are used to reduce 2 protons to hydrogen in a process that obligatory accompanies nitrogen fixation in anaerobes, making a total 8 electrons required for the molecular nitrogen. We are already discussed in previous slide now. 8 electrons, 8 protons are needed for the conversion of the atmospheric nitrogen into soil nitrogen. Okay. And next slide. I hope the slide is visible. Next slide. So the second step of the nitrogen metabolism, that is nitrification. So first one is the nitrogen fixation. So nitrogen fixation is one of the most important step of the nitrogen metabolism. Why? Because nitrogen is available abundant in the atmosphere, but it is in molecular state, which is not used by the plants. In this regard, the molecular nitrogen is conversion into different usable forms by the involvement of the microorganisms by the biological nitrogen fixation, and physical processes during the rainy season or during the raining, or even though the lightning processes, some little amount of the atmospheric nitrogen is conversion into soil nitrogen. And also the chemical processes in Haber-Bosch method in artificial manner, in the laboratory, the molecular in atmospheric nitrogen, it is conversion into usable forms of the ammonia. So the first fertilizer or first chemical is produced in the laboratory, that is urea. Okay, so that is in the process of chemical nitrogen fixation. So these 3 methods are involving in the nitrogen fixation. So that is the first step of the nitrogen metabolism. Am I clear? Then the second step is nitrification. So in nitrification, also we are observed the 2 steps. The nitrification involved in 2 steps. The first step is the ammonia is converted into the intermediate, that is hydroxylamine, that is catalyzed by the enzyme the ammonia monooxygenase. And the second step, that is conversion into the hydroxyl aminate into nitrate, and it is catalyzed by the enzyme, that is hydroxylamine hydroreductase, oxidoreductase. Hydroxylamine oxidoreductase, this is the second enzyme which is conversion of the hydroxylaminate to nitrate. So this process we can call as nitrification. So this nitrification is also depends upon the different enzymes. So that is nitrogen. Some of the bacteria also involved in this process nitrification. We are going to clearly discuss in the biological nitrogen fixation. That is also one of the most important topic we'll discuss tomorrow, biological nitrogen fixation. But the entire overview of the nitrification, which is involved in the biological process, or even though the living organism or in the physical manner. So these 2 steps are involved. So first one is the ammonia is conversion into the intermediate form, that is hydroxylamine, That is in the catalyzing process, the hydroxylamine is converted into nitrate or nitrite. So in this nitrite, the hydroxylamine oxidoreductase enzyme is needed. Then I hope this slide is visible. Next slide. Dhanalakshmi, Snehalata.
Yes, sir.
Reddy garu, Shirisha, Mitrama. Okay,
Girls.
okay, they are
Yes, sir.
enjoying because of this is the first class, na? They are enjoying. Okay, I hope you are enjoying, my dear friends. Even though respond, are you enjoying?
Girls, tell me.
No, sir.
Okay, very good. You are follow my class or not?
Yes, sir.
Following, sir.
Okay, Dhanalakshmi. Okay, very good. Then the second step, that is nitrification, is the oxidation of nitrite into nitrate. So first one is the catalyzation or catalytic process, that is hydroxylamine into nitrite. Okay, then the second step of the nitrification is oxidation process. In this oxidation process, nitrites are converted into in the presence of the oxygen. Oxidation means it is utilization of the oxygen atom or oxygen molecule. So in this process, the nitrite is converted into nitrates. Okay. So this is second stage. And third one is the denitrification. Denitrification is not useful for the plants, but also it is in the process of the nitrogen metabolism. The denitrification is the process that converts nitrate to molecular nitrogen or the atmospheric gas or gaseous nitrogen. In this process, the reaction represent the step of reduction nitrate to denitrogen gas. It is represent the complete reaction of denitrification. In this process, thiobacillus denitrificates bacteria which are involved. It is conversion of the nitrates into molecular nitrogen. Okay, so that's why it is continuation process in the process of nitrogen cycle. Okay, tomorrow we're going to discuss about the nitrogen cycle, complete nitrogen cycle once again. Don't worry. Then next slide. Then amino acid synthesis. Amino acid synthesis is also one of the second anabolic process in the nitrogen metabolism. Okay, so anabolic process, one is the nitrogen fixation, which is involved into the entire nitrogen cycle. The second step or second process of the anabolic process, that is amino acid synthesis. Amino acid synthesis is one of the most important. Why? Because it is useful for the synthesis of the different amino acids which are useful for the development of the plants and also the all type of metabolic activities. Okay, even though the osmoregulatory processes. Then ammonia thus formed as a result of nitrogen fixation is not given out. It is highly toxic and used for the synthesis of amino acids. Okay, then amino acids are the building blocks for the synthesis of proteins. Because of amino acids are the basic units of the proteins, you know. Proteins are the complex molecule, but the basic molecule or simpler molecule which are responsible for the formation of the protein by the number of proteins, by the involvement of the peptide bonds. Am I right? So the amino acids are transported through phloem to other parts of the plant for the synthesis of the proteins. The majority of the amino acids are synthesized in the plants by two main processes. Number one is reduction amination. Then second one is the transamination. First one is reductive amination. Kshayakarna amina paddhati. Amination and a paddhati. So then reductive amination, ammonia with alpha-ketoglutarate acid to form glutaric acid in the presence of enzyme glutamate dehydrogenase. Okay, so these alpha-ketoglutarate acid which are available in the process of the what we can say that the Krebs cycle or the amphibolic pathway or citric acid cycle, you know, the citric acid cycle or tricarboxylic acid cycle. Though through that cycle, the alpha ketoglutarate acid is available abundant source. This alpha ketoglutarate acid to form glutamic acid. It is one of the amino acid. And remember, my dear friends, this process depends upon the glutamate dehydrogenase enzyme. Then second one is the transamination. Okay, then transamination is one of the second step of the amino acid synthesis. Okay, then a reversible reaction, which is an alpha amino group. Alpha amino group, it is enzymatically transferred from amino acid 1 to then alpha keto acid to resulting formation of new amino acid and another alpha ketoglutarate acid, keto acid, that is derived from the original amino acid. So this pathway responsible for the deamination of most amino acids. Okay, one of the major degradation pathway which convert essential amino acid to non-essential amino acids. This step we are observed here, this is the transamination reaction. This is amino acid 1 and alpha keto acid, and enzyme is responsible for the conversion into amino acid 2 and alpha keto acid 1. Okay, so the transaminase and pyroxyl phosphate, which are responsible for the conversion into amino acid 1 to amino acid 2 and alpha keto acid 2, alpha keto acid 1. Then see here, the protein synthesis. The third anabolic process of the nitrogen metabolism, that is protein synthesis. I hope, my dear friends, you are follow my class. First one is the nitrogen fixation. The second anabolic process, that is amino acid synthesis. Then third anabolic process of the nitrogen metabolism, that is protein synthesis. In this protein synthesis, we know that proteins are made up of long chains of amino acids. From one or more chain called polypeptide chains. The amino acids bond to each other by peptides or amide bonds. Amide bonds. See here, the carboxyl group, one manaku amino acids sambandhinchiనటువంటి దాంట్లో మనకు ఏమేమి ఉంటాయి అనేటువంటిది మనం చూసినప్పుడు, when we are observed, then number 1 is the carboxyl group. Then one amino acid reacts with the amino group, and the next amino acid that is releasing a molecule of water and a result peptide bond. That is result peptide bond is formed. So this may be illustrated with the two simplest amino acids, that is glycine and alanine. So we know that, I hope you know about some of the different amino acids, methionine, glutamine, okay, valine, leucine, isoleucine, glycine. Okay, these are some of the amino acids, So these amino acids illustrated here. So this is glycine and this is alanine. Okay, and both the amino acids are going to mingle or the interact with the two amino acids, with the water molecules is released and the dipeptide bond is produced. Okay, madam, is there any problem?
No, sir.
Okay.
No, sir.
So two amino acids are going to bind to form dipeptide bond. Like that, so many amino acids are going to form long chains of the amino acids, and the forms of one or more chain called polypeptide chains. So the polypeptide chains are amino acid bonds to each other by peptide bonds or amide bonds. So this slide shows about the two dipeptide. See here, dipeptide we are observed here. So this process leads to the formation of the or synthesis of the protein or protein synthesis. Okay, so why? Because nitrogen metabolism is most important because of the formation of the proteins or protein biosynthesis or amino acid biosynthesis. And even though different metabolic, even though the catabolic processes are responsible by the major macronutrient or macromolecule, that is nitrogen. So that's why nitrogen metabolism is most important in plant physiology. So that's why we are discussed here. So tomorrow we will going to discuss about the complete picture of the nitrogen cycle, then biological nitrogen fixation, and some of the other most important phytohormones or plant growth regulators. So my dear friends, I will stop here. Regarding of this faculty exchange program, tomorrow we're going to discuss about the complete nitrogen cycle. Tomorrow, nitrogen cycle, and day after tomorrow, we're going to discuss about the biological nitrogen fixation by the involvement of the some of the prokaryotes or the bacteria, and symbiotic nitrogen fixation in formation of the root nodules in the leguminous plants. We're going to discuss tomorrow. Okay, my dear friends, I will stop here. Regarding of today's class, if you have any doubt, please ask me. I will clarify your doubts.
Yeah, thank you, sir. Students, if you have any doubt to ask, ma'am. No doubts.
Our students are
Genius.
very
Genius students.
genius students. So that's why they doesn't have even single doubt.
Yeah, thank you so much, sir. Thank you,
Okay,
students.
Manasvi, is there any doubts? Manasvi, Manasa, Ankita, Alekhya.
No, sir.
Alakya is a faculty, sir.
Alekhya, madam ji.
Good,
Hmm.
good evening, sir.
Madam, good evening. Okay, Rajani madam, thank you.
Thank you, sir. Thank you so
I'm going
much.
to wind up the session.
Yeah, thank you so much. Thank you, students.
Okay.
Thank you, sir.
Tomorrow,
Mama.
tomorrow same time we will meet.
Yeah, okay, sir. Thank you.